Good day, everyone. Welcome to my YouTube channel. I'll be discussing the basic structure and functions of antibodies. What is an antibody? Simply put, an antibody is an immunoglobulin, which is a glycoprotein, which is produced in response to a foreign material known as an antigen. And the antibody can recognize and bind to this antigen that caused its production. Serum glycoproteins can be separated in an electrical field based on their charge by movement through a gel. The gamma globulin band is relatively wide as it contains a heterogeneous class of immunoglobulins such as IgG, IgA, IgM, IgD, and IgE. Let's look at the structure of antibodies or immunoglobulins. Most antibodies or immunoglobulins are bivalent, that is, they have two combining sites. All immunoglobulin molecules have a basic structure composed of 4 polypeptide chains, which are connected to each other by disulfide bonds. Each light chain consists of about 220 amino acids and has a mass of approximately 25 kilodaltons, while each heavy chain consists of about 440 amino acids and has about 50 to 70 kilodaltons. The heavy chains are structurally distinct from each immunoglobulin class or subclass. The constant regions of the light chain and the constant regions of the heavy chain have amino acid sequences that do not vary significantly between antibodies of the same class, while the variable regions from different antibodies have different amino acid sequences. And these regions, when folded together, form the antigen binding sites. The 4 peptide chains are arranged in the form of a flexible Y with a hinge region. The hinge region allows the antibody to be flexible, enabling its combination with the antigen. The stalk of the Y is termed the crystallizable fragment, or the FC portion, and it contains the site at which the antibody molecule can bind to a cell. The top of the Y-shaped antibody consists of 2 antigen binding fragments, FAB, that bind with compatible epitopes or antigenic determinant sites. The FC fragments are composed only of constant regions, while the fragments antigen binding have both constant and variable regions. Now, the light chain can either be the kappa form or lambda form. In human immunoglobulins, the carboxyl terminal portion of all chains is identical. Thus, this region is termed the constant domain. There are 4 very similar sequences that define the subtypes. You have the lambda 1, lambda 2, lambda 3, and lambda 4 with their corresponding constant regions, C1, C2, C3, and C4, with respect to the lambda chains. The constant domains of the heavy chain form the constant region. The amino acid sequence of this region determines the classes of heavy chains. Let's look at the classes of the antibodies. In humans, there are 5 classes of heavy chains designated by the lower case of the Greek letters gamma, alpha, mu, delta, and epsilon. The properties of these heavy chains determine the 5 immunoglobulin classes. These IgG, IgA, IgM, IgD, and IgE respectively. However, there are variations in 2 immunoglobulin classes and can be classified as isotypes, referring to the variations in the heavy chain constant regions associated with the different classes that are normally present in all individuals. 2 allotypes, the genetically controlled allelic forms of immunoglobulin molecules that are not present in all individuals. And the idiotypes, referring to individual specific immunoglobulin molecules that differ in the hypervariable region of the FAB portion. Here is the basic structure of the immunoglobulin, consisting of 4 polypeptide chains. I have the 2 light chains and then the 2 heavy chains. These 4 polypeptide chains are joined together by disulfide bonds. Here you have the antigen binding sites, and the hinge here, the hinge region, gives flexibility to the antibody molecule, enabling it to either close in or widen. You have the variable region, as explained earlier, with slightly different amino acid sequences, and then the constant region with similar amino acid sequences. Let's look at the 5 types of immunoglobulin. The first one to be considered is immunoglobulin G, IgG. IgG, or immunoglobulin G, accounts for about 80% of the immunoglobulin in the immunoglobulin pool. It is the major immunoglobulin in human serum, present in blood plasma, tissue, and tissue fluids. It is responsible for providing protection against bacteria and viruses by opsonizing the invaders and neutralizing toxins. By opsonization of these microorganisms, they are marked for destruction by macrophages and neutrophils. It is also one of the immunoglobulin classes that activates complement by the classical pathway. This has been discussed extensively in one of the previous lecture videos uploaded. IgG is the only immunoglobulin molecule that's able to cross the placenta and provide natural immunity in utero to the unborn baby and to the neonate at birth. There are 4 human IgG subclasses. These are IgG1, IgG2, IgG3, and IgG4. Let's look at immunoglobulin M. Although most IgM appears to be pentameric, about 5% of the human serum can exist in the hexameric form. What do I mean by the IgM existing as a pentamer? The basic monomer, the basic structure of the immunoglobulin as a monomer, combine 5 of them together. Here you have the FC portion in the center. The bacterial cell wall antigens such as gram-negative lipopolysaccharides may directly stimulate B cells to form hexameric IgM without a J chain. IgM is known to account for about 10% of the immunoglobulin pool. It is a pentamer, as I said earlier, consisting of 5 monomeric units, each monomer composed of 2 heavy chains and 2 light chains. The monomers are arranged in a pinwheel array with the FC ends in the center, and they are held together by the disulfide bonds and a special J or joining chain. IgM is secreted into serum during a primary antibody response. It is also the first immunoglobulin made during B cell maturation and is expressed as membrane-bound antibody on B cells. Since IgM is so large, it does not leave the bloodstream or cross the placenta. IgM agglutinates bacteria, activates complement by the classical pathway, and enhances the ingestion of pathogens by phagocytic cells. Here is the structure of the immunoglobulin M. Each unit is known as a monomer, and there are 5 of them. That is why it is called IgM, is called a pentamer. They are joined together by the disulfide bond with additional joining chain known as the J chain. The structure of immunoglobulin A, IgA. IgA accounts for about 15% of the immunoglobulin pool. It is present as serum IgA as well as secretory IgA. IgA present in the serum as a monomer consists of 2 heavy and 2 light chains. Most IgA, however, occurs in mucous secretions as a polymerized dimer held together by a J chain. IgA has special features that are associated with secretory mucosal surfaces. When transported from this mucosal associated lymphoid tissue to mucosal surfaces, IgA acquires a protein termed the secretory component. Secretory IgA is a primary immunoglobulin that is associated with mucosal associated lymphoid tissue. Secretory IgA is found in tears, saliva, seminal fluid, and breast milk. In these fluids and related body fluids, it is known, secretory IgA is known to play a major role in protecting surface tissues against infectious microorganisms by the formation of an immune barrier. For example, in breast milk, secretory IgA helps to protect newborns from infections. In the intestine, secretory IgA attaches to viruses, bacteria, and protozoan parasites. This prevents adherence of pathogens to mucosal surfaces and invasion of host tissues, a phenomenon which is known as immune exclusion. Secretory IgA also binds to antigens within the mucosal layer of the small intestine, leading to the excretion of the antigen secretory IgA complexes through the adjacent epithelium into the gut lumen. This helps to get to rid the body of locally formed immune complexes and decreases their access to the circulatory system. Secretory IgA plays a role in the alternative complement pathway. Here is the structure of IgE, that there are two monomers, that is a dimer, and it has a secretory component added to it with the G chain. Structure of immunoglobulin D, IgD, is an immunoglobulin found in trace amounts as a monomer with a monomer structure similar to IgG, found in trace amount in the serum. Beg your pardon. IgD antibodies do not fix complement and cannot cross the placenta. However, they are abundant on the surface of B cells in combination with IgM and bind antigens, thus sending signals to B cells to start antibody production. Structure of immunoglobulin E. IgE makes up only a small percent of the total immunoglobulin pool, where you have the classic skin sensitizing and anaphylactic antibodies belonging to this class. IgE molecules have 4 constant region domains, C1, C2, C3, and C4 on their heavy chains. The Fc portion of the C4 chain can bind to special Fc receptors on mast cells and basophils. When 2 IgE molecules on the surface of these cells are cross-linked by binding to the same antigen, these cells degranulate. And this degranulation releases histamine and other pharmacological mediators of anaphylaxis, such as slow-reacting substance A. Here is the structure of IgD, having the basic 4 polypeptide chains. Here are some links for further reading. Thank you for listening.